Hello everyone, this is Kalpana Chaudhary, and in this video tutorial I am going to discuss about genetically engineered insulin. Applications of biotechnology are actually widely categorized, such as their role in agriculture and in medicine. The medicine that are produced using these technologies are greatly reliable, they are safe, they are pure and are more effective with less or no side effects. About 30 such medicines are used all over the world, but in India, only 12 of them are allowed. Genetically engineered insulin is one such application. And before going into the core mechanism of how this insulin is produced, let us first understand the basic characteristics and structure of insulin produced in our body. As we know, insulin is a hormone, a protein hormone to be more precise, and it is produced in beta cells of islets of Langerhans. And insulin is known to maintain the levels of sugar present in our blood by converting glucose into glycogen. That is, that insulin decreases the blood glucose level. The first question which might arise in our mind is why there a need to produce this genetically engineered insulin? Why do we need an external source of insulin? As I've already told you that insulin regulated the blood glucose level. So any deficiency in insulin will lead to a higher blood sugar level causing diabetes mellitus. In such conditions, to decline or to reduce the glucose level from our blood, or for that matter to cure diabetes mellitus, insulin from external sources is required. Initially, bovine insulin was being used. What is bovine insulin? Bovine insulin is the insulin which is isolated from either cattle or pigs. So this insulin, the bovine insulin, was initially used, but it was later found to be allergic to humans. And that is when genetically engineered insulin was developed using the gene transfer technique. Okay. So insulin in our body actually is not synthesized in its active form. Rather, it is produced as proinsulin. Proinsulin is the inactive form of insulin which needs to be activated first. As I've already told you that insulin is a protein hormone, and we know that proteins require to attain a specific structure to be functional. Same is the case with proinsulin. Proinsulin is the inactive form of insulin. So to convert proinsulin into active form, there are certain structural modifications which needs to be done in the structure of proinsulin. This here, this is the structure of a proinsulin. It consists of three polypeptide chain. This is the polypeptide A, polypeptide B, and connecting both these A and B polypeptide chains is the polypeptide C. Okay, the C polypeptide chain is actually a linker polypeptide, and its job is to bring these two polypeptides together. As A and B polypeptide come close to each other, disulfide bond between them is formed. These disulfide bonds are actually formed between the sulfur containing amino acid residues of these polypeptides. We have two sulfur containing amino acids. Those are methionine and cysteine. Once these disulfide bonds are formed, the C polypeptide is gone and proinsulin is converted into active insulin. Thus, the structure of active insulin is made up of two polypeptide chains, that is A and B. So what is the difference between the structure of a proinsulin and active insulin? Proinsulin is made up of three polypeptide chains A, B and C, where C polypeptide chain links the A polypeptide chain with the B polypeptide chain, and the structure of active insulin is formed of only two polypeptide chain A and B, and between these polypeptide chain disulfide bonds are present. Okay, so now moving forward. So what did scientists do? They isolated genes for polypeptide A and for polypeptide B. Both these genes were first allowed to transcribed into mRNA, and these mRNA using reverse transcriptase were then converted into a DNA strand known as the cDNA. Okay. cDNA stands for complementary DNA. Now now this cDNA is incorporated into the plasmid and a recombinant is formed. These recombinants are introduced into E. coli. Now what was the need for this? हमने cDNA क्यों बनाया? हमने क्यों ऐसा नहीं किया कि जो जीन हमने आइसोलेट किया है पॉलीपेप्टाइड ए और बी का, उसको डायरेक्टली प्लाज्मिड में इनकॉर्पोरेट करा के और ई कोलाई के अंदर इंट्रोड्यूस करा दिया. हमने mRNA बनाया, फिर मतलब इतना लंबा ये 2-3 स्टेप क्यों ऐड किए? So the reason is simple. As we all know that these genes for insulin genes are from us, from humans, and our host is E. coli. E. coli is a prokaryotic animal. What happens in a eukaryotic transcription? The eukaryotic gene undergoes transcription and it forms a hnRNA. hnRNA stands for heterogeneous nuclear RNA. This hnRNA ये कोडिंग और नॉन कोडिंग दोनों पार्ट से बना होता है. दैट इज इट कंटेन्स बोथ एक्सॉन्स एंड इंट्रॉन्स. बट हमारा जो कोडिंग पार्ट है जिसमें इंफॉर्मेशन है, जिसमें जेनेटिक इंफॉर्मेशन है, इज एक्सॉन. बट इस एचआरएनए में इंट्रॉन्स भी प्रेजेंट होते हैं. सो ट्रांसलेशन में मूव करने से पहले फर्स्ट वी हैव टू रिमूव दोज इंट्रॉन्स एंड दीज स्टेप्स. हाउ आर दीज इंट्रॉन्स रिमूव्ड? देयर इज अ यहाँ पे पोस्ट ट्रांसक्रिप्शनल मॉडिफिकेशन होता है जिसमें स्प्लाइसिंग और लाइगेशन जैसे स्टेप्स होते हैं. तो एचएनआरएनए में स्प्लाइसिंग में क्या होता है कि सम स्पेसिफिक एंडोन्यूक्लिएजेस आर यूज्ड व्हिच इंट्रोड्यूस कट बिटवीन दिस एचएनआरएनए एंड देन दीज इंट्रॉन्स आर रिमूव्ड एंड ligase enzyme is used to join exons together and और एक mRNA बन जाता है जिसमें only coding part है. And this mRNA is used here. Why? Because जो हमारा यहाँ पे host है वो एक prokaryote है and the process of इसमें splicing और ligation के gene नहीं है. Because the process of splicing and ligation require enzymes which are only present in eukaryotes, but the host cell used here are of E. coli, which is a prokaryotic cell, and E. coli do not have any enzyme for splicing and ligation. Thus, to minimize this error, cDNA was taken and was introduced into E. coli. A and B genes were introduced into different E. coli. That is कि हमने A जीन को एक E. coli में डाला और B जीन को दूसरे E. coli में डाला, and these E. coli synthesize and these E. coli synthesize only A polypeptide and these one synthesize only B polypeptide chain. So now these two polypeptide chain were isolated from these E. coli and were kept under condition where disulfide bonds can be formed, and this is how insulin was genetically engineered. This insulin was was called humulin. This entire experiment was actually performed by an American pharmaceutical company, Eli Lilly. Okay. This entire experiment was performed by an American pharmaceutical company, Eli Lilly. Eli Lilly introduced the genetically engineered insulin in the market on July 5th, 1983. This date is really very important, the date and year. So Eli Lilly introduced this genetically engineered insulin in market on July 5th, 1983. Later on, two pharmaceutical companies introduced an analog of human insulin called the Humulog. Okay, so the genetically engineered insulin, which was the first insulin which was formed from human, was Humulin, while the analog of insulin is Humulog. Okay, these are important. And when we talk about genetically engineered insulin, these are some scientists जिनको याद रखना is important. First is Banting and Best. What did they do? They isolated insulin from dog's pancreas, and Frederick Sanger, he gave the amino acid sequence of insulin. Okay, this was it. Thank you.